So today I'm going to talk about a little of the work that we've been doing, looking particularly at CD8 T cell dynamics in clinical biopsies that have been collected from patients with melanoma treated with combination immune checkpoint inhibitors. A lot of this work was done during my postdoctoral research in Tony Rebus's lab at UCLA. So patients with melanoma refractory to anti-PD-1 have improved outcomes following treatment. And this was shown in a 3-to-1 randomized phase 2 clinical trial recently published in Nature Medicine, showing that patients, upon the failure of anti-PD-1 therapy, should go on to receive the combination of ipilimumab plus nivolumab compared to ipilimumab alone in the secondary or PD-1 refractory setting. In this particular study, one of the secondary endpoints that we focused on was the CD8 T cell dynamics. And we saw kind of a variety of changes with respect to baseline and on therapy biopsies, seeing some patterns of increases in CD8 densities. But with the review of all of these biopsies by dermatopathologists, we were able to start considering that the CD8 T cell dynamics weren't the only thing being captured by these on therapy biopsies during treatment. We also started to see features of what we called features of pathologic response. And a lot of these patterns have been observed more in the neoadjuvant setting, describing surgical specimens to identify pathologic complete responses. But these were consistent where we would see in some responding biopsies, we would capture this CD8 T cell infiltrate. But in other ones, we would capture the necrotic tissues, some levels of fibrosis, patterns of what are called melanophages, where melanoma cells are being eaten up by immune cells, and areas of regressing tumor, which wasn't something that we really saw in the patients who were progressing on therapy. So we really just wanted to focus on this fundamental question of how do immune cells coordinate with CD8 T cells to mediate clinical responses to combination therapy. So given that we had enrolled 94 patients from across the country in this cooperative group study, we wanted to best leverage the biopsies that were collected from these 86 patients. And while we did pursue bulk sequencing, today I'm really just going to focus on the MIBI that was performed, the multiplex ion beam imaging, on baseline biopsies that had been collected following the progression of that prior PD-1 therapy. And those on-therapy biopsies that were collected after one cycle of therapy. So to pursue this study, and this was a collaboration with the Parker Institute for Cancer Immunotherapy, we took sequential sections that were collected from the blocks and underwent various histopathologic annotation using the H&E images, S100 staining for melanoma cells, and CD8 staining for T cells. And the next sequential cut got shipped to IonPath for a 32-plex panel across 800 by 800 micron regions of interest that were selected to span the entire biopsies. And we were able to collect data from biopsies at the baseline time point, that on therapy time point, comparing paired samples across patients who responded or progressed on combination therapy, as well as some patients that had progressed on the monotherapy arm as an additional comparator. Because of the audience here today, and this is building upon a lot of the really thorough introduction provided in the earlier sessions, we did take the time to modify various steps of this pipeline in order to automate both cellular and regional analyses across all of these biopsies and across these hundreds of regions of interest. And so we used a hierarchical cell typing method across 14 different cell types using those 32 markers because they were quite expansive across both myeloid and lymphoid subsets. And we implemented cell segmentation with cell typing in order to improve our challenges associated with cell segmentation. This was especially challenging given the heterogeneity of these samples that had been collected. We also leveraged patterns of regional annotation for the tissue architecture, noting things like related to the vasculature, tumor regions, and lymphoid aggregates. Not seeing too many in this particular study, but we did consider that these may be prevalent and related to clinical response. So we utilized what had already been established for the segmentation by the Mesmer algorithm from the Angelo and Van Valen labs, applying channels associated with the nuclear segmentation and accounting for various membrane markers, particularly the HLA markers and several of the immune markers, to deploy whole cell segmentation across these images. And you can see it does a pretty good job, or as best as it can, in these various tessellations. But we did take additional steps, and this was led by particularly Jessica Maxey and Zaid Bustami, in order to not only annotate those individual cells, but we performed typing across those individual pixels in order to expand those segments based upon those cells with weirder or larger shapes. And I note this because it did allow us to expand the segment areas of those cell types where we would expect this pattern, overcoming some of the challenges in segmentation, particularly in fibroblasts and macrophages and tumor cells, which tend to be slightly larger than those or irregular compared to the smaller lymphocytes that you can see didn't change very much in the size. With this regional annotation, we were able to leverage various aspects of image processing in order to identify areas of lymphoid aggregates. You can see a small region here neighboring an endothelial vessel that was labeled based on the presence of CD31 positive cells. But we were able to also go through all of these really messy biopsies and label all of the various areas that contain tumor regions based upon SOX10 positivity with melanoma cells. And so here, this whole gray area is showing those melanoma areas that were identified, as well as noting the individual tumor cells within them. And you can see some areas are more sparse than others, but giving us a general set of areas that we could compare in order to better annotate the cells involved. So there was a lot of exploration done across these various samples, and given that there could be diverse patterns across patients, and you can see varying levels of tumor cellularity, both at baseline and on therapy, we started to consider what was different and what was consistent across patients based upon their response or non-response to combination therapy. And so the first thing that we immediately saw that there was a decrease in tumor cell content. And while this felt obvious, this allowed us a quantitative way of starting to measure these levels of tumor regression. I know that there's a huge asterisk behind that given the sample heterogeneity associated with clinical sampling bias, but the fact that this was consistent and less consistent in the non-responders to either combination or ipilimumab monotherapy, we started to think that this may be indicative of that actual tumor regression consistent with how the pathologist had annotated the samples. We also saw that subtle increase in the CD8 T cells and CD4 T cells. So when we quantify those cell types with respect to the melanoma content and looking at that ratio, that's when we were able to start capturing this dynamic of putative CD8 T cell infiltration combined with tumor regression, but this was paralleled by increases in CD4 T cells, macrophages, and monocytes. And this is where we were really excited about the fact that what we were quantifying based upon this multiplex imaging panel was starting to become more consistent with the qualitative observations that were coming through from how the pathologist had annotated these images. So the next logical step with regards to these cells is, what are they doing? How are they behaving? And we wanted to start by using an unsupervised clustering approach to understand what was going on. So in order to accomplish this, we took each CD8 T cell and we annotated it based upon its total expression of each marker within the cell, as well as the percentage of each other cell type in its surrounding region. And this allowed us to account for both the phenotype and the neighborhood at the same time. Going into dimension reduction by UMAP and lighting clustering, we were able to identify 13 different cell clusters. And so this is, again, just excited to follow this path of analysis where we're able to start to see certain clusters. This is cluster 0. And shown here is the expression of CD45 rho, CD8, granzyme B, and PD1. We can start to see the expression of these things, not only on that cell in general, but polarized within that cell in the direction of the various melanoma cells that are neighboring it. And so we were pretty confident about this approach in terms of our annotation of these cells, our measurements, being able to identify these biologically relevant processes. Granzyme being effectively capturing some of the active cytolytic activity that might be coming from the CD8 positive T cell, PD1 polarized at the immune synapse, as well as CD45 Rho, which is a marker of that effective memory state in that it has been antigen experienced. So as we reviewed all of these various clusters, we noticed that the features associated with their spatial location was also coming up with regards to which features were associated with that cluster's identity. And so cluster 0 had Ki67 expression. While it's not shown in that image, it is nuclear expression on that particular cell. Things like PD1 and LAG3 and TIM3 and those various exhaustion markers, activation markers. But we also saw tumor and macrophage, giving us some indication that the things around it were also informative on what biology was happening in those biopsies at that time. This led us to the hypothesis that the CD8 T cells with shared location features also share these phenotypic characteristics. And so to that, I want to highlight the analysis strategy strongly driven by Daniel Chen in the lab, where he took the CD8 T cells and labeled them based upon spatially distinct partitions. And so with our hypothesis that these CD8 T cells may have similar activities going on at the same time, we were able to perform dimension reduction on those individual partitions, not the individual cells themselves, in order to start assessing how those communities of cells might be behaving together. So we did explore this based upon UMAP clustering and expression similar to the prior analysis, but I'm going to highlight the pseudotime analysis that I think is really telling with regards to what the CD8 T cells may or may not be doing with respect to response. And so the diffusion components associated with the pseudotime analysis created this nice little boomerang image and the 7 lighting clusters. And when we assessed the features associated with these 3 particular clusters, when in Leiden-2, we found that there was little to no expression of activation markers or putative antigen presenting cells or cooperative cells in the CD4 compartment. We also noted that these particular cells were located near the blood vessels based upon that regional annotation. And then with Leiden-1, we saw that down here at the trough of this little boomerang, we were able to see that there was increased expression of CD163 in the myeloid compartment, FOXP3 in the CD4 compartment, TIM3, but really a lack of those activation markers in the CD8 positive T cells, giving us an indication that these may be exhausted CD8s, but not necessarily activated or effector cells at that time. And then finally, over here in Leiden-0, we were able to see TIM3, CD45 Rho, and consistent with that prior analysis in the individual CD8 T cells, these entire groups of CD8 T cells were also following along this phenotype, with additional expression of M1 markers in the macrophage compartment, as well as Th1 helper CD4 positive T cells. And finally, coming back to the relation with respect to those various clinical groups, we saw that this Leiden-2 was the series of groups that did increase over the course of therapy, or there were more of them on therapy compared to baseline in those responders, but not necessarily that difference in the non-responders. The non-responders to combination therapy had slightly higher percentages of these M2 Treg and exhausted T cell populations, both at baseline and on therapy. And given that there were less dynamics in the CD8 T cells overall, it may be supportive of the fact that those are the CD8s that were there, and that didn't very much change. And then finally, in Leiden-0, we see these putative anti-tumor T cell populations, both at baseline and a little bit and on therapy, together indicating that these responders may have more of these. And then we postulate that the CTLA-4 facilitates the influx of more of these cells over the course of therapy. My favorite thing about imaging analysis is that we can always come back to the original data from which it was generated and say, do we believe it? And so given that this is a clinical biopsy, similarly annotated as before, we can label those melanoma regions by SOX10. Here you can actually see the lumen of several blood vessel areas around here. And here is one of those Leiden 0 populations with the associated monocytes and macrophages, as well as the CD4 positive T cells. And we can see even rather than just the depiction of this, the raw imaging also shows those little hubs of CD8 positive T cells reacting to those melanoma cells with relationship to the surrounding vasculature. So just in summary, this higher plexity and the added information at the protein level for the various cell types, particularly endothelial cells or SMA positive fibroblasts and parasites, really was able to help us better capture these irregular cell types. And we can even get to the subcellular resolution and consider these regional features all in parallel, depending on how we enter the data analysis. And we are excited to see that consistent with what's been done in various models, as well as suggested based upon lower plexity or lower resolution analysis, we do see these exhausted effector CD8 T cell phenotypes seem to be playing a role in mediating this response, and their co-localization with those Th1 and M1 macrophages maybe giving us better resolution of the biology that's happening there. This non-activated CD8 T cells nearby blood vessels, it's not just about where the T cells are going and what they're trying to kill, but also where they're coming from. And so that indication that CTLA-4 is mediating that process in responders compared to the non-responders can better support the mechanistic implications of these drugs in the future. But those CD8 T cells that are present in patients who don't respond don't seem to be necessarily important with respect to how they're responding. They could be stuck there, or there might just not be a good microenvironment to support CD8 T cell infiltration into those tumors. And so I think I'll get to the acknowledgement slides in just a second, but really these quantitative cell dynamics across hundreds of images that were qualitatively analyzed by dermatopathologists has allowed us to expand and feel confident about our analysis approaches across these very, very heterogeneous biopsies moving forward to understand the clinical responses. So with that, this is really my favorite slide. The SWOG Cooperative Group study started back in 2016. And so this study was primarily led by Dr. Rebus and Dr. Ari Vanderwald, but the dermatopathologist input by Dr. Phil Scampia at UCLA, very talented undergrads and postdoctoral students in the lab, Daniel Chen and Saif Bustami, as well as the many others from the statistical, technical, ends, as well as the funding agencies that have gone into this work. So looking forward to any questions that you might have.